a pleasure to be here. I came from New Zealand a few days ago, and as you can see, this is the map of New Zealand. We have a North Island, we have a South Island. We're much smaller than Australia, but we do have science and research activities. We have Auckland here, which is a major center of research and development activities, but I'm based in the lower North Island at Massey University. It's a small town called Palmerston North. Half of the population is scientists and students, so it's a rural town, but it is also a life science hub. We have a concentration of lots of life science in this particular area. We know that Australia has beautiful beaches, but we have nice beaches too. On the west coast we have the volcanic beaches, and on the east coast we have the coral beaches, the white beaches. So I'm talking about something completely different. I'm talking about the use of microorganisms to manufacture nanoparticles displaying antigens. And for this application, we have harnessed a natural capacity of microorganisms to actually to produce a reserve material. And this is a fattened up microorganisms. We have overfed that organism with sugar, and what they do is they take up the sugar and deposit the sugar as a reserve material. And in this particular case, it is a polyester, a high molecular weight polyester. And this capability to make these reserve materials inside the cell is widespread among bacteria. About 50% of the bacteria you isolate from the environment can do this. So we have looked into the pathway and into the biosynthesis routes to make this polymer and how it is made. But to make a long story short, we realized that these inclusions are actually beads, nanobeads, nanoparticles. We looked into their composition. The white core indicated here is the lipophilic polyester, and the surrounding, the boundary, is made of protein. It's a protein coat. The polyester is amorphous, it's non-crystalline, and this is our basic structure we have been looking at for quite some time, and we have been engineering to display various protein functions. And I will show you, this is the chemical structure of the polyester. The constituent in this case could be 3-hydroxybutyric acid, if that would be a methyl group, and the 3-hydroxybutyric acid is a constituent of our blood. So degradation products are very biocompatible. And this material has been already used in clinical studies as an extracted polymer from microbial fermentation. So if we look into the self-assembly process of these polyester granules within the bacterial cell, it's depicted here. We have the soluble synthase. It's only one protein actually which is critical, and that's the synthase that takes up the substrate. That substrate can be made available in any living system. It's actually coming from acetyl-CoA, and we know that's a central metabolite of any living system. I don't know life without acetyl-CoA as a central metabolite. So what happens is the enzyme starts the synthesis of the polyester chain. As it emerges out of the enzyme, the whole enzyme polyester chain complex becomes amphipathic and self-organizes into a polyester granule. The core is the polyester and the boundary is the polyester synthase. And you only need to actually put that polyester synthase into a living system and you get your polyester particles. But bacteria are the most efficient. They are prolific. They cause the high yields we are after, and they can be easily engineered and manipulated. And again, electron microscopy picture of a cell which has been overfed with sugar or any other carbon source, and we have turned on the pathway of making these polyester granules. And this is E. coli, which does not naturally produce those polyester granules. But because it's the best studied organism on Earth, we have used this organism also widely accepted for the production of biopharmaceuticals. If you do atomic force microscopy, you can look into the surface structures of these polyester granules, showing you the rough surface caused by the polyester synthase and the nice spherical structure of these inclusions. So you can also just isolate the enzyme and buy a substrate from Sigma, which is a racemic mixture, so you get only 50% utilized because it's a stereoselective conversion. And then you can make these granules in vitro, no problem. It happens quickly. You start with a clear solution. Over time it becomes turbid, you get those particles. The cell, the bacterial cell, does this 4 magnitudes cheaper from glucose than buying that from Sigma as a pure chemical. And that's the advantage of the cell factory being utilized in that process. So we have done a lot of work around this polyester synthase as it attaches to the polyester core. We have looked into the biochemistry, structure, topology of this particular enzyme, and then came up with the possibility to engineer protein functions into that protein on a genetic level without impacting negatively on the ability of the enzyme to make those polyester beads. And then you can add further proteins, further antigens, enzymes, whatever protein function you desire, peptides. You can have repetitive peptides. You can make your desired combination of protein functions at the surface of these particles. I have to go back just 1 second. The protein is anchored covalently. This is a natural covalent link to a polymer support done within the bacterial cell, and that's what we are harnessing in our bioengineering approach. So this is the enzyme, the synthase, and we know sites where we can actually manipulate the synthase to engineer those protein functions, and we have done very big protein domains within that protein without impacting on the ability to make those beads. This is the polyester synthase, and what we do is actually we overproduce it at the surface of those polyester beads. And then that's the genetic construct here. You make a fusion protein, but now we also have sites within the synthase which do not interfere with polyester bead formation. And then you can make these particles inside the bacterial cell, displaying in homogeneous orientation your desired protein. That can be quickly turned around. We do synthetic synthesis of these genes. So we get gene synthesis done by a company. It takes a few days to actually make the constructs, and then we can produce at large scale those beads, and the yields are actually quite stunning. So that's how it works. You put your gene construct, your hypergene, into the microorganism. That happens quickly, and then you get your beads made. They display the protein function of interest. And the yields are up to 80% of the cellular dry weight is polyester. So the bacterial cells are really happy to make a lot of these materials, and we do high cell density fermentation. You disrupt the cells, and then you have to clean up your beads. And we have developed a process over the last 5 years to actually get to the purity levels required for medical applications. Fermentation, high cell density fermentation, the purification of the beads. You can formulate them into powders, enhancing shelf life and stability. And given that we can immobilize, a range of proteins and protein functions, we have been looking into all these applications. We spun out a company in 2009. It's a spin-out company of the university. It's wholly privately owned, and is now commercializing the various applications from this platform technology. So we started with bioseparation applications, and now we are looking into vaccines. In particular, the focus currently is diagnostics, where we are about to register our first product. So don't read this, it's just we have done quite a few publications. We patented the technology and the applications, composition of matter and processes, and looked into diagnostics. Where can you take a bead and use the surface functionality in diagnostic applications. We have done lateral flow analysis. We have done ELISA. You take the beads with the antigen, immobilize them to an ELISA plate, and read the binding of the antibody. Or you can have actually an antibody or a binding domain directly immobilized into the beads Again, that's all coming back from that hypergene put into a bacterial cell, and then you can repeatedly produce abundantly at scale, cost effectively, those materials. Then we ended up, we did, FACS, beads, binding, sequestering antibodies by displaying antigens. But then also we came across, why not doing a skin test? We are not in humans yet, but we are in cattle, and I will share that with you. This is FACS. This is a bead. We used a bead displaying the MOC antigen, which is the myelin oligodendrocyte glycoprotein relevant to multiple sclerosis. That has been displayed. This is serum. This is OVA anti-sera. And these are MOC anti-sera. And you can see the dilutions of the sera up to 100,000. We can see still a difference between the anti-MOC containing sera as compared to the background, just as an indication. So then we came across last year, and published that last year, filed a patent, and signed a tech transfer agreement with a CMO to produce that material very soon. We immobilized antigens relevant to TB. There's an issue of cattle TB in New Zealand. It's not actually an issue, it's very carefully managed in New Zealand, and it's actually, there's an interesting story to that. The very well catered for brush-tailed possum in Australia is a pest in New Zealand, and everybody hunts it and shoots it and kills it and poisons it because there are 60 million of those possums. They have no natural predators, and they are so widespread, and they are a vector for TB in New Zealand. And the cattle comes across a dead possum, sniffs at it, gets TB, so it needs to be managed. There's a demand. The current existing commercial offering for the skin test, that's how it is being tested in New Zealand and worldwide, is an extract, a rough extract containing 1 to 200 proteins from Mycobacterium bovis. We reduced that to 3 to make it specific, because the current commercial offering is unspecific. Lots of false positives, makes the farmers nervous, costs a lot of money. And there's a lot of cattle in New Zealand. There's a lot of cattle in Europe and in Australia as well. Less cattle in Australia. In the US. So reduced it to those particular proteins, antigens with a history as being known to be immunodominant, and applied that to our beads, immobilized it, made the beads, developed the process. So now we get 60,000 tests in 1 liter E. coli culture in 2 days, produced by that process. We have formulated it so it can be nicely injected. We did dose response analysis, we did a whole range of tests, and that's the fusion protein from those beads. You can load the beads on a gel and you see the protein, and then you can inject it into cattle. We have now tested 30,000 cows in New Zealand with very good results, 4 times more specific than the commercial offering, cost-effective production. We are now looking into the human market as well. So the veterinary market is about $350 million, and the human market, is expected much bigger. Now taking the same, almost the same bead prototype into the blood assay. There is an interferon gamma release assay, which is a second tier, more specific assay for cattle TB, but also that's the same equivalent for humans actually. It's all equivalent. Now we get all these animal trials done, and we would like to move into developing diagnostics for humans. Then we looked into a range of antigens to be displayed onto our beads. We used different bacterial hosts like Lactococcus lactis with 1,000 years of food safety history. It's in yogurt. You eat it every day, not every day, but occasionally. And then E. coli with a history being used for the production of biopharmaceuticals. So we made antigen-displaying beads, published that in the past, injected that into mice, and then we saw first proof of concept. There is a cell-mediated immune response as well as a humoral immune response, and went further to challenge studies, and I will show you a few examples of that. We have been looking into vaccine candidates for new disease targets, so we are always on the lookout for new targets. You have maybe knowledge of good antigen combinations. You desire to be combined onto a particle and being cost-effectively, abundantly produced. We can quickly turn that around and test that. also interested in other delivery routes than only subcutaneous or parental delivery routes. So example one is the Mycobacterium tuberculosis on polyester beads. We took the famous EZ6 and AG85A and went on to obtain proof of concept. And here you can see that's the IFN gamma release. And I should actually explain here the x-axis where we have the beads displaying the antigens, and this is the antigen amount compared to the free antigens, the recombinant proteins. And as you can see here, we have an antibody response, and it's significant over the control, and there's a significant increase also compared to the recombinant free proteins. So we have a little bit of antigen sparing here, and we also see significant responses in terms of IFN gamma release, indicating a Th1 response. Further cytokine profiling also indicated that we see a Th17 response using those TB antigen in mice. And in blue, these are the beads from E. coli. In green are the beads isolated from Lactococcus, from the yogurt bacterium. And they resemble each other. Then going into challenge experiments with Mycobacterium bovis in mice. Mice immunized with the E. coli beads displaying those antigens and the Lactococcus beads in lung showed a significant reduction of the M. bovis load, and that is compared with the current gold standard, the BCG, indicated here. And then we looked into spleens as well and saw, particularly from the E. coli beads, there is a reduction in spleen. And then we looked into the uptake by dendritic cells with those beads displaying the antigens. These are dendritic cells, no beads present. In the presence of beads, we can see uptake. This is electron microscopy. After 10 days, the dendritic cell is really fully packed with those beads. They seem to love to take up these polyester beads efficiently. And we looked at the dendritic cells and looked at the maturation caused by the beads in general. It's independent of the presence of antigens. The beads in general cause maturation of the DCs. And that's also coinciding with the release of interleukin 12, which is of course then linked to the Th1 response. So what we can conclude from this first example is the beads have been produced which display these mycobacterial antigens. Immunization with these beads initiates an immune response, and it is Th1 and Th2 and also to some extent Th70, and we saw protection. Next example is by just adding capsular polysaccharides to those beads and see if there's any synergy. And we looked into meningitis and pneumonia, did the standard cross-linking as has been FDA approved for the commercial offerings, and just monitored the cross-linking efficiency, and the recovery is very similar to the commercial yields obtained. So the conclusion was here that we can actually successfully conjugate those polysaccharides to the beads, and then also could confirm the identity of the capsular polysaccharides as being displayed on the bead surface. And here you can see the beads with the capsular polysaccharide mediate a humoral immune response. These are the doses here: first dose, second dose, third dose. And if you are, and that's interesting, if you look at this, that's the positive control, the commercial offering. But if you look at this column here, if you just take the polysaccharide not linked covalently to the beads, then you don't get the immune response. This is a negative control. And the same for the other capsular polysaccharide. We don't see that immune response. So apparently the co-delivery of beads and the antigen are critical. And we checked the quality of the antibodies and saw a superior quality of the antibodies in terms of opsonophagocytotic titers. So the killing ability of these antibodies obtained from the beads were superior to the commercial offering indicated here in green. We did not do the CPS from Neisseria meningitis yet. Example 3 is HCV, where cell-mediated immune response is critical as well. We took the famous core antigen, all informed from the literature in collaboration with a group in Cuba. We looked at the immune response in mice, and we did challenge experiments using the recombinant vaccinia virus. Again, these are just the wild-type beads without antigens, just the naked beads, and these are the beads with the core antigen. And this is ELISpot to look at the IFN gamma release. And you see a significant increase when you have the antigens present here. And then in the challenge experiment, we had 60% of clearance when mice were challenged with the recombinant vaccinia virus expressing the core antigen. And then these are the wild-type beads without the antigen, so of course we don't see the reduction in viral titers. And so we're currently looking into other TB antigens to make a better TB vaccine, hopefully. And then the Johne's disease is interesting to agriculture. Sheep is affected by Johne's disease, then meningitis pneumonia, where we have these serotypes, variety of serotypes where current vaccines are not providing full coverage. We heard about the 13-valent vaccine on the market for pneumonia, and then hepatitis C, dengue, and Pseudomonas aeruginosa are currently being looked at. And I should mention that we are also in the position to engineer the pathway of polyester particle production into the pathogen. So the pathogen can actually make the particles. And some pathogens actually do their own particles. So it is out there, it is natural, and the polyester is natural. So that it is intriguing to us to follow up on that. Of course, we can engineer anything into E. coli, Bacillus subtilis, Lactococcus, and we can arrange we can make multiple epitopes and then put in a domain of an interesting antigen, a vaccine candidate. We can load up that synthase with large domains of proteins and still get particle production. It's a very robust system. So I'm about to finish. I don't actually want to read all of this. The polyesters are actually FDA approved, and they have been in clinical trials. And I'm just talking about antigen sparing here, enhanced shelf life. We can make powders which can be reconstituted. We are in the field, so we have a bit of safety. 30,000 cattle are all happy. Of course, they are bigger than humans, but they're all happy. And we have, in case of the core antigen, the hepatitis C core antigen, we used only 2 doses as compared to a commercial mix of antigens under development. So we are reducing doses by immobilizing the antigens on the surface of the beads. And last but not least, without the dedication of my co-workers who are working day and night, not only spending time on the beach, but enjoy the work. It's an international group at Massey University, and some of them are employees of the company. So we have a company lab, and we have a research lab. We have basic science being translated into applied and commercial science. And we have Price Buttle, who is driving, the skin test reagent development. He is a standing in TB for a long time, and have good funding from the government and industry funding. And thank you for your attention.
I love the idea of the antigen and the vehicle being produced in the same bacteria. But in terms of, I saw that you were looking at using dengue antigens. Some viral antigens don't express well in bacteria. What antigen are you targeting for dengue?
We're looking at the envelope protein, D3. We use a truncated version, and we use the serotype which is relevant to Asia, the Asian serotype. So we have to, that's correct, we have to engineer proteins, in particular if there's post-translational modification requirements. Pathways have been engineered in E. coli, but they are less efficient. We also use endotoxin-free E. coli strains these days as well. But in some cases we have to engineer the epitopes into the system to allow efficient expression. That's correct.
Using E. coli for, you showed different possibilities, including protein expression. And as everybody knows, a major problem with protein expression is the aberration with folding inclusion bodies. So, did you try with your system to see if exclusively inclusion bodies protein can be expressed also?
So what we found, and we looked at that very closely, we found inclusion body formation and polyester particle formation are mutually exclusive. So you get the synthase, as soon as the synthase domain becomes active, it starts to make the polyester particle and keeps the protein separated on the surface of the polyester particle. No. If you tamper with the synthase and make that synthase weak, so it does not form particles, you go the other pathway. But if you get particle production, it excludes proteinaceous inclusion body formation.
And does the synthase by itself make inclusion bodies overexpress it somehow?
No, it doesn't form protein inclusion bodies. It starts to make the polyester, and that separates the proteins. It does not allow them to aggregate.
So now, if you want to separate your protein, is there a way to clean the protein from the synthase?
Yes. We did that as well. We published that, but we are still working on that possibility because it is actually a protein production system as well. It's not only displaying antigens, but you can also produce antigens by having like self-cleavable tags, like inteins. You lower the pH, and then all of a sudden the proteins release, because we can obtain those polyester particles easily. So you get a very fast and quick purification of those particles. Then you release the protein of interest by self-cleavage, or you can use proteases, specific proteases, of course. And we are currently working on a very high-yielding specific system, which leads to protein production which otherwise would form at that level of yield proteinaceous inactive aggregates, inclusions. Yes.
We showed the dendritic cell taking up a lot of the particles.
Yeah, it's a long time. So
What pathways are triggered in the dendritic cell? So how does the adjuvant, what's the mechanism of the adjuvant effect?
That's a good question. We have done only currently little work in that regard. We would be keen to follow up on that. What we know is the maturation of these dendritic cells. We know the IL-12 release is happening, but we really don't know about the fate of what's happening to the adjuvant, what's happening to the polyester over time. That's something we need to look at to understand the mechanism. Next stage, yeah. Couldn't see a difference from the experiments we have done so far.
Is there a time lag? There's nothing like that.
It's yeah. It's the same currently from the limited experiments we have available. Just limited by data, so we have to get more and more data. It's all data driven, isn't it?
Okay, it's a good time to finish the session. I'd like to thank all the speakers.